everyone, and thank you for joining us for today's webinar entitled Strategies for Improving Soluble Protein Production in E. coli. My name is Karen Klayman, and I will be moderating the event today. Okay, just a few items before we begin. Please use the chat window at the left side of the screen for entering any questions you may have. The dialog box should be set to be viewed by all. We will then address all questions at the end of the presentation. Also, during the webinar, we will be asking the audience to participate in 3 interactive polls, which is an opportunity for all of us to share our experiences with recombinant protein production. Your answers are anonymous, so please feel free to participate. Use your cursor to select all of the options that apply, and then see the responses appear live in real time. Thanks for participating. Okay, I am very pleased then to introduce today's speaker, Beth Fry, who is the product manager here at Lucigen. Here you go, Beth.
Right. Thank you, Karen. And thanks to all of you who are joining for today's webinar. We are very excited to have you with us as we talk about improving protein expression in E. coli and how to get more soluble protein out of your expression experiments. And for today's webinar, we're going to start by reviewing a little bit of the history surrounding recombinant protein expression in E. coli, and then we'll talk about some challenges that researchers face when expressing recombinant proteins. And finally, we'll move on to talk about some methods and products that you can use to overcome those challenges. So let's first talk about why we overexpress recombinant proteins. We know that recombinant proteins are used for both commercial purposes and to answer basic research questions. And expressing a recombinant protein inside a cell allows us to study the protein's function. For example, identifying other proteins it interacts with, or when and how the protein is expressed, or what function it may have in a signal transduction pathway. And purifying that recombinant protein and studying it in vitro allows us to determine the protein's structure and to study, for example, enzyme kinetics or reaction conditions or reaction products. And we can also produce abundant amounts of the protein for use in biotherapeutics and industrial enzymes or for research products and services. And here are some examples of those applications. So we have a protein crystal structure here which requires purified protein, and we've used human kinases as an example to represent how recombinant proteins can be used for drug discovery. These are a huge family of over 500 enzymes used in both in vivo and in vitro assays for enzyme activity along with binding and inhibitor assays. And this is rituximab, which is a biotherapeutic monoclonal antibody, which is used to treat non-Hodgkin's lymphoma, leukemia, and rheumatoid arthritis. And then for an example of research tools, we chose restriction enzymes, which are purified proteins that almost every molecular biologist is familiar with. And industrial enzymes like lipases and proteases can be used in consumer products like detergents and other cleaning solutions. So now let's talk about E. coli as a host for recombinant protein production. According to a 2014 search of PubMed articles, over 70% of recombinant proteins that are expressed in E. coli, and that's a trend that continues throughout the years. And this is due to unique advantages that E. coli offers, namely the well-understood genetics, the ease of cell transformation with foreign DNA, high-density cell growth in relatively inexpensive media, and the ease to which the growth can be scaled for large-scale protein production. However, the workflow of expressing recombinant proteins in E. coli is not without challenges, as we all know. And so let's talk about these general challenges. And this is an overview of an example workflow which starts with cloning and proceeds to expression and protein purification and finally to analysis. And some of the challenges are listed below each step. And we won't name each of these challenges now, but I imagine one or more of these roadblocks are familiar to most researchers who are expressing recombinant proteins for any application. And there are enough potential challenges listed here to spend an entire day talking about them, but for today's presentation, we're really just focusing on the first 2 steps, cloning and expression in E. coli. So let's talk a little bit more about those steps. So our cloning step starts with choosing an appropriate expression vector. A lot of factors affect this decision, as we'll see in a moment, but after the expression vector is chosen, the gene of interest is inserted, and the resulting clone is then transferred into an E. coli host strain for protein production. The protein is then expressed, and the success of these steps is then determined by evaluating protein yield and solubility and/or activity. So let's break down that first decision, which is choice of vector. The choice of promoter can be a big factor. Would you like an inducible or constitutive promoter? The selection marker and the replication origin are all items that can really affect the end result during your evaluation. So we really need to think about the entire workflow when we think about vector choice. Now the next step is inserting the target gene, which can be done in many ways. And on the left, you'll see a cartoon representing the classical PCR cloning workflow in which the vector of choice is digested and dephosphorylated, and then the insert is amplified and processed. Then there's a ligation step followed by transformation. And many other cloning technologies can be used as alternatives to this classical workflow, and some of them are listed here. And they all vary a bit in terms of workflow and time requirements, but the desired end result is a target gene inserted into the expression vector. And then the next step is transfer of that expression vector into E. coli. And there are a couple of different methods that can be used. Chemically competent transformation can be used with chemically competent cells, and electrocompetent transformation can be used with electrocompetent cells. And both of these methods have their advantages. Chemically competent cells are a bit easier to work with and don't require any special equipment, but they generally have a lower transformation efficiency. And electrocompetent cells have a higher transformation efficiency, but they do require an electroporator. They also, though, have a slightly faster workflow. So depending on your needs and the available formats of your desired host stream, you could use either method. And then the last step that we talked about is inducing protein expression. And here is where those choices that we made earlier on in the workflow are evaluated, essentially. And success here means that the target gene is expressed at the desired level, and that it's soluble, and most of all, that it's active and functional in your assay or in your downstream application. And we listed some of the variables that can help determine success here. Or they could develop into roadblocks or parameters that require optimization. So when we do have challenges with protein production, what really happens? Let's dig a little bit deeper into what's going on at the molecular level. And what we see is that transcription and translation are coupled in the cell. They're happening at the same time. And that's shown here by transmission electron microscopy, and then by the cartoon below that kind of illustrates what's going on in the figure above. So RNA synthesis starts in one direction, making mRNA from the DNA. And as the mRNA is transcribed, it's also being translated. The ribosomes are binding and translation is happening. So we have a coupled transcription and translation going on in the cell. And when we're overexpressing proteins, we often have more artificially active promoter regions, and we're working with exogenous DNA or DNA that's foreign to the host. And some of the issues we may see are low TNR availability due to rare codons that are used in a sequence, or a large amount of secondary mRNA structure. We could have a non-optimal translation rate, or even just too much protein expression for the cell to handle. And these problems can lead to very poor results, like degraded proteins, improper folding and insoluble protein, or even toxicity, which affects cell viability and can even cause the culture to die out. So these problems really are a problem for people doing protein expression. And here is a table that lists some of these problems and the source of the problems that we talked about, and some potential solutions for these issues. And I'm not going to go into all of this in detail. There's a lot of information here, but just note as you sort of skim this that the solutions to these issues span the entire workflow we just discussed. So in order to implement some of these solutions, you have to go all the way back to the cloning step, and in some cases even to the design of your DNA sequence. So some of those solutions are very time intensive and can really stop or even reverse the project of your project process. So here are some solutions that we're going to talk about today in the context of challenging applications that you may have. And we'll start by talking about how to clone faster in a method that is truly high throughput compatible. We'll talk about how to express high levels of proteins and how to control that protein expression level, as well as how to improve protein solubility. And we'll also talk about a solution for successfully expressing membrane proteins in E. coli, which, as you know, membrane proteins are a particularly difficult class of protein to express in a prokaryotic system. And that overview will bring us to our first poll question. I'll hand it back to Karen to manage the poll. And just as a reminder, if you are watching the webinar in full screen view, please hit the escape button to switch out of full screen in order to see the poll question and take part in the voting. Karen?
All right, thank you, Beth. Here is our first poll question. What is the native source and type of the protein you are trying to express? And again, please select all the options that apply to your research. All right, go ahead and select all the different options. We have bacterial, plant, mammalian, or synthetically designed sequences for the source. And for the types, membrane proteins, enzymes, transcription factors, antibodies, or antibody fragments. All right, for the native source, it looks like it's basically prokaryotic or bacterial proteins, mammalian, and enzymes are the primary type of protein. Wonderful, thank you, everyone. We'll conclude the first poll. Thank you, Beth.
Hey, thanks, Karen, and thank you, everyone. I hope that was as fun for you to vote as it's fun for us to see the results, and we really appreciate your participation in our polls. So as we discussed, there are challenges spanning this entire workflow that we talked about. And the first step is almost always a cloning step. So let's start with the cloning challenges. Cloning in general is a very time-consuming multi-step process. We talked before about the classical PCR cloning workflow, which had many different steps, and each one of those handling steps is an opportunity for something to go wrong. So it's hard to automate or to scale a multi-step process as well, where you need lots of pipetting and lots of sample manipulation. So I'd like to introduce you to a simple, easy, and high-efficiency cloning method that helps solve some of those challenges. This is Expresso cloning, and it's a simple method for cloning your PCR product into a lucigen expression vector. And it takes advantage of the natural homologous recombination pathway in E. coli to fuse together the insert and the vector. And how can this help you? Well, it's definitely faster. There are no post-PCR cleanup steps that are required to purify your PCR amplicon. And there's also no enzyme or ligase incubation steps either. So it's fast, directional, and it also introduces no cloning scars or extra DNA sequences into the vector. So Expresso cloning also uses pre-processed linearized vectors, which have extremely low background, and that means that the cloning method is very highly efficient. And in this gel showing 18 colonies that were screened by PCR for correct insert, 17 out of the 18 showed the correct insert. So that's well above 90% efficiency. And our R&D scientists here use the Expresso method extensively, and we routinely see efficiencies approaching 100%. So how does it work? Well, let's talk about the steps. There's really 3 simple steps. First, amplifying your target via PCR. And the PCR primer should include 18 base pairs of overlap with the Expresso vector sequence. You then add your PCR product and the Expresso vector to competent cells and proceed with transformation as you normally would. It's a chemically competent transformation method. And that's it. The homologous recombination occurs inside the E. coli cell, which generates your expression clone. So in comparison to traditional PCR cloning, it's faster. It doesn't require any extra reagents like enzyme mixes, and it doesn't require any enzyme incubation steps. And so it actually decreases the handling and pipetting steps significantly. And this makes this technology very useful for both day-to-day cloning, as well as making it an attractive method for cloning on automated platforms, which is one of the challenges we talked about earlier with methodologies. So I mentioned the PCR primer should be designed with an 18 base pair overlap with the Expresso vector, and this slide shows how easy that really is to implement. And on the top we see a partial sequence of a linearized Expresso cloning vector with the insert site right here. And below that is the sequence of a PCR product that's designed for cloning into the Expresso vector above. And you'll see here are 18 base pairs of overlapping sequence on the 5' end and over here on the reverse complementary end of the sequence. And below that is an example of the forward primer sequence. So this sequence would just be added to the 5' end of your gene-specific sequence on your primer. And so 18 base pairs of Expresso vector sequence, 18 to 24 nucleotides of gene-specific sequence. And the same on the reverse, except you're going to use the reverse complement sequence. It's that easy to design these primers. And now, the Expresso systems are complete kits for protein expression, and by that I mean that the kit includes almost everything you need to clone and express your gene of interest. So they include linearized, dephosphorylated Expresso vector. It's pre-processed to have very, very low background. It includes competent cells for both cloning and protein expression, and a control insert that can be used as a positive control for cloning and for expression, as well as any protease-mediated cleavage if the vector includes a cleavable fusion tag. And it also includes vector-specific primers for confirming the sequence of the clone. We also include any sugar solutions that might be needed for protein induction, and the protease enzyme for tag cleavage if the vector contains a cleavable tag. And this description is going to bring us to our second poll question. So I'll hand it back to Karen again for the poll. And again, please remember, if you're watching the webinar in full screen view, please hit the escape button to switch out of full screen so that you can see the poll question and participate in voting. Go ahead, Karen.
Okay, second question. Which challenges are you facing in your protein expression projects? Again, please choose all that apply to your research. And we have low or no expression, low solubility, toxicity, inactive protein, other issues, or no issues. Please make your elections. All right, we have problems with solubility and low or no expression. Some are just inactive protein, so it's expressed but it's inactive. All right, great, thank you. Low or no expression, low solubility lead the pack here. That will conclude the second poll. Thank you.
I see we had one person who said they had absolutely no expression challenges and that their expression was always perfect. That's wonderful. We're happy to hear that. But for everyone else who may be experiencing problems, we can talk a little bit more about solving those particular problems. So we have talked about the cloning challenges. Now let's move on to solving some problems here in the protein expression realm. And these problems can occur at transcription, or at translation, or during protein folding. And here are some of the problems that we have chosen to focus on here at Lucigen because we feel that they are some of the more common problems that people experience. So remember, as we discussed before, the choices made about expression vectors and host cell lines upstream really affect the results here. And so what can we do upstream to help prevent these roadblocks from occurring in your workflow? So the Expresso T7 cloning and expression system can help to solve some of those expression challenges. The vector includes a T7 promoter for high-level expression. And to control toxicity, we use high control cells to reduce basal expression, and we'll talk more about high control in a bit. We can also help to increase protein solubility with an optional SUMO tag. And the gene of interest is inserted with Expresso cloning to save time through your workflow. And we just went over Expresso cloning a few slides ago. So let's look at the Expresso vectors themselves. So this is a system that is optimized for high-level expression and tight control of expression level. There's a choice for either N-terminal or C-terminal 6-his tag, and an optional N-terminal SUMO tag as well. And the vectors are optimized for protein expression. They're very small in size. They're only about 2.2 kb, and that's very small for an expression vector. You'll also find, as in many Lucigen vectors, that we've used the kanamycin selection marker instead of ampicillin to help reduce satellite colonies during cloning. And we've also inserted transcriptional terminators here on either side of the insert area to eliminate any unwanted transcription into and out of the cloned fragments. So unwanted transcription activity can be a source of clonal instability. And because of that very low background we talked about, because of these very high efficiency vectors and using that high-efficiency Expresso cloning system, it's going to really help your workflow in terms of time savings, and it's also going to help you control leaky expression. So it can also be used for expressing toxic targets, and this is due to that high control technology that I mentioned before. So these are high control cells, and they differ from normal BL21 DE3 cell lines. So in a normal BL21 DE3 line, both the T7 RNA polymerase and the target gene are regulated by promoters that have this LAC operator. And in an uninduced state, the LAC repressor binds to the LAC operator and represses transcription of the TNA7 RNA polymerase and the target gene. However, with high copy plasmids that are used for protein expression, sometimes there just isn't enough LAC repressor in the cell to sufficiently suppress both the T7 RNA polymerase and the multiple plasmid copies of the gene of interest. And if there's leaky expression of the T7 polymerase, that can drive leaky expression of the target gene. And if it's a toxic protein, that can be very deleterious to the cell. So high control cells solve this problem by introducing higher levels of the LAC repressor to provide greater control over gene expression. And this results in less toxicity and in higher cell density achieved before induction, which can mean a large difference in soluble protein yield. And the T7 Expresso kit includes both a high control 10G line for a cloning strain to avoid any deleterious leaky expression during the cloning step. And it also includes high control BL21 for protein expression. So because of the high expression levels and good control over expression, this is a great system for both routine and for toxic targets. Some expression vectors like PET28, which is shown here in this comparison, PET28A, contains a LAC repressor and a separate promoter on the plasmid backbone. And that's actually one way to get around the problem of leaky expression due to too little LAC repressor in the cell. Just put another copy on the plasmid backbone and increase the copy number. However, keep in mind that the LAC repressor does make the plasmid much larger. This PET28A plasmid is around 5.3 kb without an insert, whereas Lucigen's petite vectors, as you remember, are around 2.2 kb. So there is a significant size difference there. In this SDS page gel comparing the soluble protein expression, on the left-hand side, we see the DNA polymerase, which is a toxic protein, and in the middle, we see a control fluorescent protein. And then on the right-hand side here, we see a membrane protein. And in each case, the stable expression of the recombinant protein is basically equivalent for both systems across those target classes. So it really is a great system for both routine and toxic targets. Now let's move on to talk about how else we might solve the protein toxicity and solubility challenges using Expresso Rhamnose cloning and expression system. And in this case, we have the benefit of a tunable promoter, which allows precision control over the protein expression levels. This level of control can help with a variety of issues, not only toxicity of protein, but protein solubility and low expression levels as well. And this system offers the same time savings and workflow efficiency advantages as the T7 system by using Expresso cloning, and it saves even more time because the cloning and expression can both be done in the same host strain. There is no need to transfer the clones to a different strain of E. coli containing T7 RNA polymerase or a DE3 strain for protein expression. It's simply just not needed. So this system has the advantages of T7 in terms of vector choices and size and the engineering of the vector. And it also includes an optional SUMO tag to further enhance protein expression and solubility if you wish. And one of the key improvements of this system over the T7 system is the promoter I mentioned. So this is a Rhamnose inducible promoter. Induction is determined by carbon source. So when the cells are grown under glucose, expression is repressed. And in the absence of glucose and in the presence of Rhamnose, protein expression is induced. And the responsiveness of the promoter system to different concentrations of Rhamnose is really key for expressing toxic targets. So how does it work? Well, this gel shows how responsive the system is to varying concentrations of Rhamnose in the growth media. Under glucose repression, leaky expression is essentially eliminated here. We see almost none of the protein of interest. in the absence of glucose and Rhamnose, there's virtually no expression either. Maybe we see a little bit of a hint here, but essentially there is none. No detectable expression. However, when the cells are exposed to a gradient of Rhamnose in the growth media, we see a concomitant increase in the level of protein expression. And this is true for Rhamnose levels between 0.001% and 0.2%. So it's very, very sensitive. And this is a really powerful thing for very toxic proteins because you can adjust the level of Rhamnose in the media to find that cell's level of tolerance for expression of that particular toxic target and then induce expression at just under that level to maximize your yield without killing your cells. And in addition to the workflow advantage of cloning and expressing in the same cell line, we can take advantage of this responsiveness to provide a hands-free auto-induction protocol. And this is where glucose and Rhamnose are provided in the culture media, and the cell will preferentially grow on glucose to a higher density while tightly repressing expression of the target protein. And then when the glucose is depleted, the cell switches over to Rhamnose and protein expression is induced. And this is what autoinduction looks like. So depending on the amount of glucose added during the initial and autoinduction setup, the protein expression can happen at an earlier time point. So here we see it at about 8 to 10 hours really showing up with a lower concentration of glucose. Or with a higher concentration of glucose, it takes longer for the cell to deplete the glucose in the media, and we get a later induction, a later switch over to the Rhamnose. So this can help increase overall protein yield, and it can also help increase the cell's tolerance for toxic protein targets. And this is going to bring us to our third and final poll question. Again, please remember, if you're in full screen view, hit the escape button, please, to switch out of our full screen view in order to see the poll question and take part in the last opportunity to vote. So I'll hand it over to Karen.
Okay, so which fusion tags have you used to express your proteins in E. coli? And again, pick every option that is applicable. His tags, DST, MBP, HA, mix, leg, SUMO, other, or none, you do not use fusion tags. His tags are a very common tag, as we can see, 84%. Flag and GST seem very common as well, and great to see. All right, everyone put in your last vote? All right, polyhistag is the winner. Thank you. That concludes the last poll.
All right, Karen, thank you. And thank you, everyone, for participating in our polls. That's really interesting that 6-his tag appears to be definitely the winner there and the most popular. So the next challenge we want to talk about is related to purification and different fusion partners and tags. And we want to tackle protein expression and solubility. So the Expresso T7 SUMO cloning and expression system contains the same advantages as either the T7 or the Rhamnose promoter systems we just talked about, depending on which promoter you choose. And it also contains an N-terminal SUMO tag. So the SUMO tag is a yeast protein. It's small, ubiquitin-like modifier, and it's about 100 amino acids. But it's known in the literature to enhance both protein expression and protein solubility. And with this system, the other nice thing is that native proteins can be recovered with cleavage of the 6-his SUMO fusion tags using the SUMO Express protease. So how do we purify these native proteins? This is just a slide showing you exactly how the workflow proceeds for purifying native proteins. So after protein expression and purification of the recombinant protein on a IMAT column, like a nickel column, the 6-his SUMO fusion is cleaved using a 6-his tagged SUMO protease enzyme. And this cleaves the tag and leaves the native target protein. Then we can do subtractive IMAC over a second nickel column to allow the target protein to flow through, and the 6-his SUMO protease and the 6-his tagged SUMO tag are both retained on the column. And this is a pretty system, but just to note that the cleavage buffer here for doing the SUMO protease cleavage does contain DTT, which can be incompatible with some nickel resins, and so we recommend testing the cleavage buffer on your nickel column prior to applying your cleaved protein in order to determine whether or not it causes nickel to leach. And if it does, if it's necessary, you can always dilute the cleavage reaction to decrease the DTT concentration prior to column loading. But this is a great way to purify and recover your native target protein in a few simple steps. So we'll talk here about some examples where SUMO tag has enhanced both protein expression and solubility of some difficult targets. And these are results of some large-scale expression studies to identify hydrolase enzymes from Fibrobacter succinogenes. Sorry. I can't talk this morning. So 48 genes were actually selected for expression trials, and they were cloned into to our petite T7C his vector. And approximately half of those clones produced soluble active hydrolase protein, which is a great start. And others were expressed in an insoluble form, which is not so great. But 5 of those genes that produced insoluble proteins were then cloned into the petite SUMO vector. And for 4 out of 5 of those genes, the soluble protein yield was significantly increased, as you can see here in this table. And the gel for 2 of these proteins is also shown. The T up top is for total protein, and the S is for soluble protein. So in the case of gene 1, without the SUMO tag, there was very little overall protein expression, and very literally no soluble protein. And for the SUMO tag version, we had a big increase in the amount of expressed protein and an increase in the amount of soluble protein as well. And then for gene 2, the SUMO tag enhanced the solubility of the target. So we see almost nothing in the soluble lane here without a SUMO tag, and here we see a nice band there. So we've talked a lot about solubility. And how do you really evaluate protein solubility, and what can you do if you do have poor protein solubility? Well, here's a common method for evaluating protein solubility by SDS page. And the protein is first expressed in your E. coli culture, and the cells are harvested by centrifugation and then sonicated to lyse the cells. And then the lysate is separated by centrifugation into soluble and insoluble fractions. And then samples of the total and soluble and insoluble fractions are run on a gel to find out where your expressed protein is and which fraction it is. So hopefully you have great expression levels in your total protein lane, which is a T, and you have it all in your soluble lane and hardly anything in your insoluble lane. That would be great results. However, if you have a protein gel that looks like this, where the level of expression isn't very high to begin with in your total protein lane, and you also have a lot of it in your insoluble fraction, then you really need to optimize something in your procedure. And there are some options for you to consider here listed at the bottom. So one option would be to purify the proteins as inclusion bodies. But the solubilization and refolding procedures are really plagued with problems, and it requires a very high degree of skill in order to get those to work, especially at scale. You can also try to optimize protein induction parameters, but that is very time consuming and it may not work. And another solution is to use an optimized system that can enhance expression and solubility with a panel of fusion partners. And that's the system that we're going to talk about next. So the Expresso solubility and expression screening system can help solve a host of issues from toxicity to expression to solubility. And it utilizes some of the same strengths as the systems that we've just talked about, but it makes it even easier for you to use. And we'll talk about that a little further here as we go along. So this system contains 7 different Rhamnose inducible expression tags, expression vectors, with an N-terminal his tag, followed by a solubility partner and a TEV cleavage site. And the fusion partners are listed here. There are 4 that you probably know of. So there's a SUMO tag, an MBP tag, and a GST tag. And then we have a control vector here with just the 6-his affinity tag. It doesn't have any solubility tag. And that is intended as a baseline for expression and solubility. And then we have 4 different novel tags. So AFV, SLY-D, TSF, and beta-lactamase are our 4 novel tags. And this is the only system that we know of that contains a panel of fusion tags. Not 1, but 7 different tags that you can test all in parallel. And all the tags are cleavable with selective protease. And it's also, selective protease is also a 6-his tag to allow easy removal. And these fusion tags can be tested in parallel with a very quick, easy workflow. So this is what the workflow looks like. And first, one set of PCR primers is designed to amplify your target. And then the gene is amplified once, and that single PCR product is cloned into all 7 of these Expresso vectors in parallel, plus the control vector. So 8 cloning reactions. And after this transformation and the cells are plated, this all happens on day 1. On day 2, you can start your cultures for protein expression. And remember, because this is a Rhamnose promoter system, the cloning and expression can be done in the same E. coli host strain. So then the following day, you're already evaluating protein expression and solubility and see if any of these tags worked for you and for your target. And following that, the protein can be purified and the tag can be cleaved with selective protease, and then you're left with soluble native protein. So this really allows you to go from gene to protein in as little as 4 days. And this system offers some real advantages for high-throughput protein purification labs. So both the cloning and the protein expression steps are minimized, and they're simplified, and they're done in parallel, which makes it a great candidate for automated platforms. And again, the cloning is highly efficient. So as shown here, we have 24 clones, colony PCR screened from Lin28 being cloned into the PSOL vectors, which are in the kit. And all of them were the correct clone. So again, this is what we usually see at Lucigen when we use this cloning method internally. So of course, you'll probably want to clone, or you'll probably want to confirm your chosen mini preps by sequencing, and or you want to confirm your expression clones by mini preps and sequencing. And you'll want to do that before scaling up or taking it to the next step in your workflow. And these higher efficiencies also mean that you can perform fewer mini preps and fewer sequencing reactions. So all through the workflow, it really reduces pipetting steps, it reduces handling, which really makes it amenable to scaling. And the system really allows you to quickly identify which tags work for your protein. So this is an example of SMAD, which is a protein involved in TGF-beta signaling, which was cloned into all of the vectors included in the T7 solubility and expression screening kit. And we're looking at a total protein, soluble protein, and insoluble protein. And we're looking at it here by SBS page. And the control clone, which is constructed with just a 6-his tag and no other fusion partner, acts as a baseline. And what this shows is that virtually all of the protein is expressed in the insoluble fraction here. So looking at the lanes marked T for total protein, we can see that SLY-D and TSF and SUMO and MBP all showed increased levels of expression compared to the control. However, when we look at the lanes marked S for solubility, we can actually see that MBP and TSF showed the greatest increase in soluble protein production. And as another example, we have GH1, which is a form of human growth hormone. And again, in the control, what we see is very little soluble expression. But in this case, there were 4 tags here in green that really increased expression levels and solubility levels. And we need to keep in mind that because we can't necessarily predict which fusion will work best on any given protein, the best tag really has to be determined empirically for each individual protein. So if you just take a minute and think about how much work it would have been to test all of these tags at one time, or to test each tag if it was on a completely different expression vector. So if you started with, say, an MVP tag on a PMOL vector backbone, you go through your cloning and your expression, and say it didn't work for your target. Then you decide to test a SUMO tag in a PET vector backbone. Then you'd have to restart. You have to re-amplify your target and re-clone it. And in this system, all of these tags can be tested in parallel in a matter of days. And the results are very clear. It really gives you a decision point to move your project forward. So if you do happen to find that you're working with a group of proteins that have similar properties, we are very happy to narrow the scope of the system for you. So you can test all 7 fusion partners. And if you find that particular fusions work well for your targets, we can provide the pre-processed Expresso vectors with the tags that you want at larger scales for scale-up or high-throughput protein expression and purification projects. So all you need to do is just contact our customer service team or contact me, and we can work with you to provide the fusion tags that you need for your project. So now let's talk about membrane proteins. We're going to switch gears just a little bit. And we know that these are extremely difficult to express in prokaryotic systems. Overexpressed C41DE3 and C43DE3 are 2 mutants of BL21DE3 that allow production of membrane proteins from vectors containing T7 promoters. These cell lines are highly referenced in the literature. They have well over 350 publications and growing. And here's a figure showing the transformation success rate, the post-transformation cell viability, and the protein expression levels of a group of 28 membrane protein constructs, all driven by T7 promoters, that were transformed into C41 and C43, and in gray, the BL21DE3, just an unmodified BL21DE3 strain. And the overexpressed strains outperformed unmodified BL21 in every category that was measured. So if you have a membrane protein that you would like to express, which strain should you try? Well, there are 2 strains that really differ in their tolerance for different proteins. So there is a list on Lucigen's website with its literature references for each strain, and it includes the native organism and the class of the protein that's expressed. So this guide can help you with your choice that you would like to start with. Or Lucigen also offers a combo pack of C41 and C43 cells, both with and without the key lysis antigen. And so all 4 cell lines. And it allows you to try your membrane protein in all 4 strains and determine empirically which one works best for you. So we've talked a lot about the decisions that can be made at the start of a protein expression project that can help mitigate some of those challenges that we see at various stages of the workflow. And here are some general tips that may be useful no matter what cloning or expression system you're working with. And I won't go into everything in detail this morning, but I did want to mention a few things in particular. So most researchers sequence verify their expression clones and then transfer into a separate expression host strain, if you're not using our Rhamnose promoter system, that is. And if you happen to be running into problems with your expression projects, one of the things you might want to do up front is to sequence verify the target that has been isolated from the expression strain, to make sure that there are no mutations occurring in that expression strain after transforming the expression strain. And we also recommend sonicating the cells to evaluate total protein expression instead of resuspending in SDS page loading buffer. So without sonication, the overexpressed protein can actually entangled with the residual DNA from the host cell, and that makes a very viscous sample that may not enter the well and may not allow the target protein to even be visualized on the gel. We see this all the time. And these results are very deceiving because it looks like there's no protein there, and it actually is. It just didn't make it into the well or make it into the gel. So if you sonicate, though, that breaks up those protein DNA complexes and allows a true visualization of total protein. So there's some other very useful tips here, but I really wanted to mention those 2 that we found very helpful here at Lucigen. So in summary today, we've talked about some of the specific challenges faced when expressing proteins in E. coli and about the specific needs listed in this table, such as expressing toxic proteins and improving solubility. And we've talked about how researchers can save time and effort by making the right choices about expression vectors and host cell lines while thinking ahead to the purification strategy and the downstream application. And we hope you found it informative and interesting. And if you'd like to learn more, here are a few resources and application notes that expand on the topics that we've discussed today. And they're available on our website at the links described in this slide. And all of the slides and the presentation today will be made available online for you to review as well. So again, we've enjoyed you with us very much, and we hope that this information will be useful to you in your research. And with that, I will hand the reins back over to Karen, and we'll be happy to take any questions about anything we've covered here today. Karen?
Okay, thank you, Beth. And so audience, please use your chat window on the left if you have questions for Beth. We'll take questions now. And in the time being, we have Husta has a question regarding does the protein of interest really remain soluble after cleaving off the SUMO tag or any other solubility tag?
That is a great question. And we do know that in some cases, you can, and not just with SUMO tags, but with other tags, you can get what's known as false solubility. So it's a case where the protein appears to be soluble with the tag, and then as soon as you cleave the tag, it chunks out of solution and becomes insoluble. And there are some things that you can do to help mitigate that. You can look at different purification methodologies. You can look at different potential buffers that you're cleaving in. We can try to optimize around that to try and keep the protein soluble after cleavage, but it's really a very protein-specific solution. You can also try a different tag, so something like the Expresso solubility and expression and screening kit might be a great way for you to test different tags if you do find that you're experiencing that false solubility. Great question.
Yes, all right. Marianne has a question about sonication. What sonication buffer do you use?
That is a great question. I don't know off the top of my head. I know that it contains DTT, and I believe it's tris-based. Those are pretty common combinations. I believe that there is a recommended sonication buffer that's in our product manuals. But if not, if you want to contact our tech support team, then Karen and our tech support team can help you figure out a good buffer to use. We have some that we use here as a standard basis. And if that information is not in the kit manuals, then we can get it for you.
Great, thanks. And Jason just came up with a question. What is the conditions that you used for sonication?
That is a great question. And I think it depends based, in all the gels that I've shown today, some of them were done on a larger scale and some of them were done with an 8-well sonication tip. So we can absolutely get you those conditions as well. If you want to contact tech support with your information, we'd be happy to help you out with the conditions that we've used. And if there's a particular figure that you're wondering about the sonication conditions for, we can get that information to you as well.
Okay, great. Thanks, Beth. And we have a question also about inclusion bodies. Are there any methods to avoid it, a part of changing the E. coli strain?
Well, that's a great question. Inclusion bodies are a tough one, right? Protein solubility is a very challenging thing to overcome. And there are some different methods. So some of the things we talked about today would be to modulate the expression level and hope that by maybe decreasing the expression level with something like a Rhamnose promoter, you can get the cell to tolerate and correctly fold that protein. Often, decreasing that expression level allows the cell to kind of catch its breath and catch up with the protein expression level if you've got a really high expressing promoter. So that's one option, is just decreasing overall expression using something like the Rhamnose promoter that's very tunable. And you can also try those solubility tags. So those really help to overcome some of those N-terminal problems that can lead to protein solubility issues. So you can absolutely try a different E. coli strain as well. That's definitely another thing that people use as a troubleshooting method. But I think maybe something like the Rhamnose cloning and expression system would be a great place to start. That would really allow you to modulate the protein expression level.
And in regards to expression levels, how does the highest, this is from Rob, how does the highest level expression from the Rhamnose promoter compare to the highest level expression from the T7 promoter?
Sure. So the T7 promoter is going to be the highest expression level that we can get, really. The Rhamnose promoter is close. If you have it all the way up at 0.2% Rhamnose for induction, it's going to be very high, but it likely will not be quite as high as the T7 promoter. So T7 is going to be highest, then the Rhamnose, but we definitely get the ability with the Rhamnose promoter to be tunable, whereas the T7 promoter is on or off, right? There's really not a gradient there that you can achieve. So if you're too high with the T7 promoter, it's going to be it's going to be difficult to modulate that, where you can use the Rhamnose promoter to modulate your expression. And I should also mention that here at Lucigen, our R&D team and our production teams use the Rhamnose promoter preferentially over the T7 promoter for just those reasons, actually. So great question.
And Jason, yes, you had a question about the slides being available. Yes, these slides will be available in PDF format on our website. So please look at the site for those files. Any more questions? Please go ahead and enter it in the chat box. We have just a couple more minutes here.
Great questions.
Yes, very good questions. I agree. All right. Looks like we've exhausted our possibilities here. Let's go ahead and conclude. Thank you, everyone. Oh, we do have one more. Is SelectTev present in the final purified protein when using the Expresso solubility kit?
Oh, that's a great question. So SelectTev is used for the cleavage of the TEV fusion tag, and it is a 6-his tagged protein. So the workflow that we talked about with the SUMO express protease, where we can remove it easily by subtractive IMAC, by applying the cleavage reaction, essentially just putting it over another nickel column, and the tag, the cleave tag, and the SelectTev enzyme will stick to the column, whereas your native protein can be purified from the flow-through. So no, if you put it over a different, another IMAC column, then the SelectTev does not need to be present in the final purified protein. You can purify native pure protein in a few simple steps, just like the SUMO express protease workflow.
Okay, any more questions? All right, with that, I'd like to thank our speaker, Beth Fry, and you, the audience, for joining us today. If you do have any additional questions, please email our technical support team at the email address on the screen. And thank you again to everyone. The webinar is now concluded.